Let me now start. Yes. Good evening. My name is Sushmita Mandal. On behalf of the German Consulate General Kolkata, I welcome you all to the fourth web-based session of our science lectures. These lectures aim to further strengthen the collaboration between Germany and India on science and research. Today, there will be two lectures. The first lecture is How Microbes Overcome the Plant Immune System will be by Professor Gunther Doehlemann. And the second lecture, Parasites Are Smarter Than Humans, will be by Professor Hemanta Majumder. Professor Pulak Sengupta will be the moderator of this session. And now I hand over to the German Consul General in Kolkata, Mr. Manfred Auster, for his introductory comments. Mr. Auster, please.
Well, thank you very much, Ms. Mandel. Good evening, India. Hello to Germany. I'm very happy to greet you on this, my first participation in this series of science lectures that have been already running for quite a bit with the German consulate here. Actually, we started. I'm aware of. We started those in 2016 already, and ever since, Ms. Mandel has been the organizer for these things, and I would very much like to praise her for that, and also mention the fact that she was the first one to contact me once it became clear that I would come to India, which was rather on a short notice kind of thing. And from then onwards, there was a possibility to see that even in Corona times, we have a good audience and that we have the possibility to reach out and foster the relations between scientists and researchers in India and Germany, something that is not new to our history. I think we share quite a bit of that, not only over decades, we can already speak of centuries in that regard, because there was quite a bit of an exchange even before the First World War between the two countries, and intellectuals and researchers and scientists, but also on the side of the humanities, there was quite a bit of an exchange between the two countries. And so I'm really glad to see that this is continuing and that we have so many distinguished fellows of the DID, but also of the Humboldt Foundation, who are still working together and are continuing to do so and spreading the message. We also have quite a lot of young researchers and scientists who want to go to Germany and have applied for that. The numbers are rising. And at this juncture, I would just like to use the opportunity to basically apologize for the fact that we had to close the visa section for about 10 days because of a Corona case. But rest assured that everyone who had an appointment will be contacted as of Monday and will get a new possibility to come and get their visas processed as soon as possible. People who have had an appointment will not have to go through the whole process once again. I know that this was one of the concerns among the student community here. We are really very happy and very proud that so many people are interested to come to Germany to do that. We're doing the utmost that we can continue and foster these relations. Therefore, without much further ado, I would like to hand over. I'm only a humble lawyer to the people who are much better positioned to speak about the topics of today. But I'm looking forward to that, and I'm really keen to see what I can take from your lectures, professors. And so I would like to hand over to Professor Pulak Sengupta from the Department of Geological Science at Jadavapur University, who happens also to be a fellow and an alumni from the Alexander von Humboldt Foundation and the DID for that matter. He is the recipient, I'm told, and here I have to look into my notes for that, the Shanti Swarup Bhatnagar Prize, and congratulations to that. And as if that is not enough, he is a proud, I hope, a proud father of a daughter who is a researcher who works in Germany together, or actually under Professor Doehlemann. So here the circle basically closes itself and shows in a nutshell how good the relations between the two sides are. Congratulations. Thank you very much. The floor is yours, Bulak.
Thank you, Herr Auska, our honored speakers, Professor Doehlemann, Professor Majumder, and my dear audience. Before I turn to my audience, I just quickly make an expression from the bottom of my heart. This year, I am ecstatic that two German colleagues of mine have awarded with Nobel Prize in Physics and Chemistry. And this is almost a repeat of 2017 when the two Nobel Prize went to my German colleagues in the same subjects. So I am really ecstatic. I hope that all the recipients of German fellowships in India, they feel the same way as I do. Now, turning to my audience, dear audience from Kolkata, different parts of India and elsewhere in the world, I, Pulok Gupta, welcome you to these episodes of Science Lecture organized by German Consulate General. And I am really very happy and really very happy because we have with us two very eminent life scientists who will be talking on microorganisms. And before I introduce these two speakers, I like to make some points for the audience. Please cooperate with us with following this request. First, you put your microphone in mute mode so that unwarranted noise doesn't disturb the speaker. The second part is very important. Please do not upload anything during the talk. This will be a big distraction. The speaker won't be able to identify that whether this thing has been uploaded. Then we have to stop, and then again the speaker has to restart. And the third point, I'm sure that once we have this talk, you may have questions or queries to these two eminent scientists, and certainly you will shoot your question, but only through the chat box. And please remember that we will accept or take this question as much as possible because we have a time limitation. We'll try to cover most of your questions. But please do not forget to write the speaker to whom your question is addressed to. Otherwise, it will be very difficult for me to know, which question should go to which speaker. So with these few things, and I just introduced our first speaker. Before going to that, before I upload my things, I must say that our first speaker, as Shushmita has already mentioned, that first speaker will be Professor Gunther Doehlemann, and the second speaker will be Professor Hemanta Majumder. Incidentally, both of them have been my next door neighbor in the sense that last 2 decades, I have been visiting the Department of Mineralogy and Geochemistry of University of Cologne, and Gunther's department only were a lens away. Similarly, I am working in Jadavpur University, and Indian Institute of Cultivation of Science is also a road away from Jadavpur University. So in that way, I am very close, both mentally and geographically, to both the speakers. Now, let me formally introduce the first speaker. I should just a moment. Can you see it? Has it come? Okay, so our first speaker is Professor Gunther Doehlemann. He is from CEPLAS. It's a big name which is abbreviated to CEPLAS, from University of Cologne. By by training, he is a biologist who did his PhD from University of Kaiserslautern, Germany in 2006. Interestingly, when I was having a chat with Professor Hemanta Majumder, he was telling me that looking at the CV of Gunther, that he could be my son, because he did his PhD in 2006 and Professor Majumder did his PhD in 1975, if I can remember correctly. So anyway, but he's a very, very, eminent scientist in his own field. And then he did his PhD, postdoctoral at Max Planck Institute for Terrestrial Microbiology in Mahabu, where he became a research group leader in 2009. He joined this department, University of Cologne, as professor in 2014. At the moment, he is the director of his biology department. Now, his main focus, among many other things, is or has been the microbial interaction of maize to understand how pathogen microbes interfere with the plant immune system. Professor Doehlemann, is an accomplished scientist. He received the prestigious Julius Kuhn Prize, and also he was awarded with European Research Council Consolidator grants in 2017. I was going through the Google, and I request you to have a look at his Google page. He is a real good scientist in his own field. Today, he will be talking on how microbes overcome the plant immune system. Meine Liebe Gunther, herzlichen Willkommen. The screen is yours.
Yeah, thank you very much, Pulak. Thank you, everyone, to this kind invitation. I'm really happy. I'm also happy to see some of you again, healthy. And now I'm trying to share my screen, and I hope that works. What do you actually see?
It hasn't yet come.
It hasn't come?
Oh, now it has come. Now it has come.
What do you see? Because I don't see something what I'm presenting.
No, you won't see. I mean, you can only see your presentation.
Okay, good. Then I have to... Okay, so now do you see a full screen of my presentation?
Yeah.
Okay,
yeah,
good.
full screen.
Very good. Yeah, so thank you very much again for this nice invitation, and to see you again in health. It's a pleasure to me to talk a bit about our science, and maybe of note to Pulak. So this morning we started the day with a literature talk that was given by Priya Mehta. So I was listening to your daughter, and now you will listen to me. The title of my talk maybe to some of you sound already quite specific. So how microbes overcome the plant immune system, but I will try to do my best to give you a bit of glimpse why this is generally relevant and what that actually means. And then of course also come a bit to that, what we are actually doing in our own research on that. So let me start very general. So we are living in a difficult time, and we're having a hard life, some of us. This year is Corona, but also plants' life is hard, and plants are exposed to many threats. Many of them are abiotic threats, like rain, temperature, wind. They're threatened by pollution, by humidity or drought, or by too much or too less nutrients. That all affects the health of plants. However, there's one big other component, and this is the interaction of plants actually with other organisms. And these interactions can be beneficial on the one hand in mutual symbiosis, but, and this is what is interesting to us today, is they of course can be detrimental in pathogenic interactions. And here we have to say that fungi actually are under attack by organisms from all class of kingdoms. So what you can see here is a plant that is infected by tabac mosaic virus. So we have virus infecting plant, and actually TMV was the first virus at all that was characterized. We have bacteria that infect plants. Here you see Erwinia amylovora. This is fire blight disease, which is actually disastrous to many food trees. And if plants are infected with this disease, usually the only thing is you can just burn them down. Then of course we have animals. Well, we have apparently big animals, like these pretty cows here who are eating plants, but more relevant in terms of pathogenesis are of course parasites like worms. You see these nematodes which infect and feed on plants, and we have many insects which of course eat the vegetative part of the plants. We have also, and this is on the bottom right, other plants which feed on plants. So here you see, for example, monotropa, and you see that these plants are not green, and they are not green because they don't do photosynthesis by themselves, because they just suck the nutrients from the plant. And then, and this is the biggest group, and this is what we're talking mainly today, is eukaryotic microbes. So higher organisms, but microbes. And here you have one big group of microbes, which is here. You see this rotten potato. These are microbes which are actually more related to plants, and they infect plants. And then the other big group of eukaryotic microbes are, of course, the pathogenic fungi. You see two examples here, Aspergillus, which is infecting maize plants. And a good friend of mine, this is the organism where I did my own PhD on, Botrytis cinereus, the so-called gray mold fungus. And I guess everyone has seen this more often in his life. So this is actually eating all kinds of fruits and vegetables you can imagine. So I mentioned now that there's two big groups of eukaryotic microbes which are infecting plants. And to visualize this a bit, I put here this colorful tree of life. What you see here is basically the phylogenetic relationships of the different branches of the kingdoms in life. You see here in green the plants, and you see here the ophistocons in this color, and you see here, by the way, do you see my mouse?
Yes, we do.
Okay, thank you. That's good. So you see here the animals, and you see closely related to the animals are actually the fungi. So fungi are much closer related to animals than to plants. And here we have plant pathogens like this powdery mildew. But then on the exact opposite side of the tree of lives, we have the heterocons. We have here brown algae diatoms. We have the malaria parasites in these groups of organisms, and we have the oomycetes. And these are actually forming very similar structures and infecting plants in a very similar way, although evolutionarily not related at all. So one example I want to give here, and this is I think everyone should have known at least once in his life or her life about this organism. This is Phytophthora infestans. Phytophthora infestans means the plant destroyer. This is an oomycete microbe that comes originally from Southern America, and this is where, of course, the potato comes from. And the history is that in the 16th century, the potato came into Europe. And then there was kind of a happy time until middle of the 19th century when the potato became the prime food in Europe, but there was no pathogen on it. But then it happened in middle of the 19th century that with a contaminated ship, Phytophthora infestans came into Europe and caused devastating epidemics and disease. And this, particularly in Ireland, caused the Great Irish Famine from 1845 to 49. And within only 10 years, more than 30% of the population dropped. You see in some areas, within just 10 years, more than 30% of the population was down. And this is mainly to starvation, but also to immigration into the United States. And this is a very nice example to see this impact of a pathogenic plant microbe on on society, because this organism is the reason why Irish people became the biggest white European immigrant group into Northern America, and still are one of the biggest groups still. So however, I mentioned that the biggest group are fungal pathogens. So why is that, or how is that looking? So what you see here is from a paper from a few years ago that was published in Nature, where they were covering actually new diseases which appear on the world. You see that particularly in Northern America, there's a lot of new animal diseases coming up. But there's, of course, more plant diseases, and this is in Europe, but also you see here in India, there are many more new plant diseases appearing. And when you look who's causing these new diseases, then you see that almost 3 out of 4 new diseases are actually caused by fungi. And currently, this makes that we are losing 10% of our global harvest to fungal diseases. So this is basically food for like 700 to 800 million people every year, which we are losing to fungal diseases. And an example for that, and also to show you how this looks like, this is a very, very common old pathogen. This is Porcinia graminis. This is the wheat stem rust. More than 2,000 years ago, the ancient Romans, they had a god, Robigo, which they had to actually protect them from this disease. They were sacrificing foxes to this god to be protected from stem. And this shows you how old this is. And in 1990, it really maybe actually so I don't hear anything. Do you hear me?
Yes, we do.
Okay. Sorry, I don't see you at the moment.
No,
so I'm asking, and it was quite soon. Okay, so this is actually a very recent, urgent example of how these diseases spread. But also, I don't want to share you just bad news with you. I just want to share you a bit my fascination on the beauty of actually these kind of interactions. And here I just prepared 2 small examples, and that I think is the beauty of flower to get infected. And another beautiful example is Microbotrium, which it's infecting Violacea. So this is a dianthus, and this forms this nice plants flowering here. And this is a healthy flower, where this one here is a diseased flower. It looks very similar, but instead of the plant pollen, you will have fungal spores inside, which then are spread. And I'm showing you these 2 examples to give you a glimpse of how sophisticated these microbes can interfere with the plants, that they can completely disturb and basically hijack the plant development to even form other organs or use the organs of the plant to spread. And this, of course, asks, let's say, much more basic questions. And one of these basic questions is, before we can understand how this actually happens, is how actually a plant can get sick, and why a plant can get sick, and why is it infected. And that means, why is this little tree here actually killed by a Phytophthora, while all its brothers here around are staying healthy? And the other question, and this is the one which I will focus on in the next minutes, how actually is a pathogen defined? 99.999% of all fungi around will not be able to cause disease on a barley plant. But this one here, Plumeria graminis, is able to develop this super highly specific infection structure and cause disease on barley. So how is this defined, and what is the basis behind that? There's, of course, multiple questions, but I will go into one of them. And one answer why this is and how microbes can do this is one certain class of proteins which they have. And these proteins which break the immune resistance of a plant is what we call effector proteins. And effector proteins basically shape the virulence of many plant pathogens. And this is one of the first and seminal pictures that has been made in a publication 9 years ago. And what you see here is a cell, and here basically 2 examples of cells that are infected with the rice blast fungus, Magna Porcio rizae. And the green thing you see here is the hyphae, and the red thing here is actually a protein which is an effector, and this effector is secreted from this hyphae into the plant cell. Yeah, and here this molecule is doing something. It is altering kind of the structure and the function of the host cell in order to cause disease. And this is basically the molecular background, basically how microbes can do that. So how to study something like this, and to do really functional analysis and understand how microbes can do this and how this works on the molecular level, what you need is actually a good model. And there it comes a bit to what we are doing, because we are studying one class of fungi, which actually are, after the rust fungi, the second biggest group of plant pathogenic fungi that exist. And this is the so-called smut fungi. We know about 1,200 different pathogens with more than 4,000 host plants. And you see here some typical disease symptoms in wheat or in oats caused by Ustilago nuda, Triticea, or Avenae. So basically, these fungi infect all relevant crop plants we have. And the beauty of them is that they have a yeast stage and they have small genomes, so they're good organisms to study in the lab. And there's, of course, one big model. And the big model organisms, which we are also studying in the lab, is Ustilago maydis. Ustilago maydis is the corn smut disease, or the common smut disease. And the symptoms, what it's causing, and maybe if you have been in a maize field once, you have seen that. This is these galls, or called tumors, which are formed in the plant instead of normal maize flowers, or here in this maize crop. And in these tumorous structures, the fungal will grow. And the beauty is here that we have a very good genetic model. We have a lot of genetic tools, and we have the most important genetic tool, which is CRISPR-Cas, to modify the genome of this organism. So a bit more about this and its life cycle. So as I said, and this is the beauty why I'm a molecular biologist, and what we want to do is we want to do genetics on our organism. And therefore, it is very good that this organism can actually grow in haploid cells, which grows as yeast. And that's actually something interesting also. These yeast cells, you will find them in nature very often, but not as pathogens. So these yeast cells are completely harmless. Only if on an appropriate plant surface, two of these yeast cells, which recognize each other and which are compatible to each other, they will fuse. And they will fuse a filament, and this filament actually starts to be a pathogen. And here you see a microscopic image, how this then punches the surface of the leaf, and it grows inside the leaf cell, and then it starts proliferating in the leaf. And this is, again, live cell imaging, so this is laser scanning microscopy, and you see here in red, the fungal cell, which is growing inside here in green living plant cells. And this is surrounding the fingers growing around the plant nucleus. And this is actually the miracle, which is this shows this intimate interaction between the parasite inside the plant cell and the cell, which is perfectly happy. And this is very, very exciting, because the plant, like we do have, they have an immune system, and this immune system recognizes initiate an immune response, but in this case, apparently it doesn't. And the reason why it doesn't is because of the effector proteins. And so, and then it grows the spores, which you can see here, and which are then released tubes. So more about these effectors. What are they and how do they look like? So I told you in the beginning that these effectors can be secreted kind of into the plant cell. And to visualize it, I showed you here another laser scanning micrograph. What you see here in blue is the cell wall of a plant cell. And this is like laser scanning microscopy. It's just a slice through a cell. Here you see a fungal cell. This is growing into this plant cell. And this is expressing a red protein. So the cytoplasm of the cell is red. And then you see a very faint green layer. And this faint green layer is one of these effector protein, which is labeled with a green fluorescence protein. So this is where these effectors go to. They are secreted into the interface between the fungus and the plant cells, and sometimes they even can go into the plant cell itself. And this is what we're looking at, because these molecules are making a pathogen to be a pathogen and make it being able to disease in the plant. So I will give you just an example on how that looks like. That was actually the first of these effectors which at that time, I analyzed by myself in person. I found that when I was a postdoctoral fellow at the Max Planck Institute. And that was a very lucky shot because that was the first effector I selected from the genome because of some features. I just liked it. I don't know exactly why that was retrospective. It is a very small protein that has nothing special on it. It has a signal which makes it to be secreted. For the biochemists, it might be interesting that it's 4 cysteine residues which form 2 disulfide bridges forming an iron knot, and then it has some glycines in the C-terminal. And if you look where it is, you see that, like the other one before, it is secreted by a fungal hyphae into the plant cell. But why is it interesting? Well, if we do genetics and if we take this gene, which is encoding for this one single effector out from the fungus, then you see here the fungal white type causing disease, causing tumors. The fungus has completely lost its virulence. So if you look in detail, the fungus will grow on the plant surface. It will try to penetrate the plant surface, but then it's blocked. And if you look how it's blocked, then you see actually, again, this is another microscopy. You see here an infection structure, and you see here the fungus growing in the plant. And here you see this mutant, which does not have this single effector. And what you see here is plant immunity. This is a plant defense response. This is particles which are transported from the plant cell to the site of infection, forming a physical barrier. This is what we call a papilla, a cell wall fortification, because this fungus is not able anymore to press the system, actually block, and it cannot be infectious anymore. And this just because of one single protein. And at that time, we spent a lot of effort to understand what it does. And actually, we found that this is inhibiting peroxidases, and therefore it prevents the accumulation of oxygen, of reactive oxygen, which is actually a trigger for this immune response. So this is basically the biochemical reaction behind. And what we found then is that these kind of effectors are highly conserved amongst fungi that infect various different plants. So you have smut fungi infecting all these different crops, but also in dicots, like this is a polygonum smut. And everywhere, if you look in the genome, we find this sequence highly conserved. And even more, if we put the genes from all these other fungi back into our maize pathogen, we give it back the ability to cause tumors. So in other words, we have here a highly conserved virulence factor, and this is something we call a core effector, which is suppressing plant immunity. And this is one of the enzymes, or one of the, it's not an enzyme, one of the proteins that makes a microbe to be a pathogen. Well, and I will not bore you with all the details that came, and just make you aware that we learned from effector studies that effectors actually come in hierarchy. So we have core effector like PEP1, they are conserved. We have a next level. We have effectors which are very specific to a certain pathogen. And you see here a sequence. Only thing you have to see that this is not similar because these are host specific. And these are also virulence factors, and they have nice functions. For example, this one here has a function to be a molecular mimicry. Just one figure here. Normally in the plant we have proteases. These proteases have substrates which they cleave. But this effector actually mimics a substrate. It will bind to a protease and will just block it. And since this protease is important to activate the immune reaction, this effector blocks the immune reaction just by mimicking a substrate of a protease. So this is molecular mimicries in the effector level. We have also effectors that actually act very specific on certain tissues or even cell types. So what you see here is a journal cover with a picture which we made, And here you see 3 maize cells. These are so-called bundle sheath cells. And in these bundle sheath cells, they are green because they are producing DNA. And this is triggered by one single effector, which we found to be active just in this particular cell type. And we later then found that actually in different cell types in the plant, the fungus expresses and activates different effectors which have certain effects, and together they activate the processes, for example, to trigger the formation of a tumor in a plant. And then, of course, the last thing is that there are many effectors. Well, we simply don't know what they're doing. And that's actually the honest answer, is that for about 90%, we have no idea right now what they're doing. And that is also one reason why we are now changing a bit the strategy. And this is the last thing I want to mention, or to show you how we are doing this. And this is a new type of approach. And this is actually using the new techniques we have. We are living in a time where we have much more genetics and genomic tools. And we're using this now to completely deconstruct fungal virulence. We are using basically CRISPR-Cas genome modification together with all the knowledge we gained in the last years to dismantle the pathogen and take away all these different effectors which they have to make it basically apathogenic. And then what you have then is a chassis. It's a chassis, it's a microbe that is disarmed and is not able anymore to infect a plant. And then we can come to the second step. And the second step is to reconstruct this. And this is basically the vision here, to establish synthetic plant biology. Well, it's a bit of personal opinion. I think we have decades or times of eras where different kinds of scientific approaches are dominating. And I think we are now entering the time of synthetic biology, which will have massive impact, not on science, but only on our social life. And this also implicates that we are establishing synthetic plant pathology, which means that if we really want to understand something, we have to reconstruct it. So that means we're using the new tools we have to really build up new sets of virulence, and also to do this for such important epidemics causing pathogens like rust fungi, where this was not possible before with tools. Until now, we have them just quite recent. And that is actually the vision for the next years. That we can only understand something if we are able to build it. And that's what we are going to do. And with this, I want to thank again you. But of course, I also want to thank the people who are involved. That's mainly our lab, but that's of course also our collaboration partners, and that's all the funding agency from the DFG, the Alexander von Humboldt Foundation, and the European Union, which is heavily funding our research. Yeah, thank you very much.
Thank you, Gunther. Wonderful. Ausgezeichnet. You presented it so lucidly. I was getting a feeling as if I am seeing a stealth aircraft that dodges past all the air defense systems and bomb the target. And well, we have a few questions. Please stay with us till we finish with our second speaker. Please stay with us. My next speaker, just a moment, I possibly lost. Has it come?
Yes.
Okay. So our next speaker is Professor Hemanta Kumar Majumder. Hemantoda, I hope that there is no mistake in your surname. No Z is J is fine.
Yeah.
Okay. Professor Majumder is presently Nazi senior scientist, Jubilee Fellow of CSIR Indian Institute of Chemical Biology, Kolkata, and he's also the adjunct professor of Department of Biophysics, Molecular Biology, Bioinformatics, University of Kolkata, Calcutta, Kolkata. And by training, Professor Majumder is a biochemist, and he has an illustrious career. But before that, I just take out a few leaves from his past. He did his master's and bachelor's from University of Calcutta, Kolkata. Then he did his PhD from Bose Institute, Kolkata, and then did his 2 postdoctorate, one from Albert Einstein College of Medicine, New York, and University of Zurich. He is a very distinguished scientist, and he has made many fundamental contributions. I will name a few. One is biochemistry of DNA, topoisomerasis of Leishmania. Both the words, I mean, it sound like German words. It's a big word. Anyway, and with reference to development of therapeutics. His outstanding contribution towards development of diagnostic and therapeutics against the deadly disease Kala-Azar is internationally well acclaimed. Professor Majumder has received many accolades nationally and internationally. I will quickly mention a few of them. He is the fellow of all the 3 Indian Science Academies. He is a J.C. Bose Fellow, National Fellow, and Rajaramanna Fellow as well. And he is also a member and fellow of Third World Academy of Science, and he has strong connection. Sorry, I'm moving perhaps too fast. He has a strong connection with German institutes as a visiting professor. He visited Robert Koch Institute at Berlin and LMU at Munich. His 2 daughters, if I'm not mistaken, are staying in Germany and doing their research. Probably they are in the job as well. And his talk will be on parasites are smarter than humans. The title of the talk is little scary for me. If the parasites are smarter than humans, then what will happen to us? But this is on a lighter note. Hemanta, this is an absolute pleasure to invite you to give your talk. Now the screen is yours. Well, before you talk, let me tell you that your PPT will be uploaded and monitored by Professor Ranjan Ganguly. And since we have a lot of audience already, and there can be a time lag of few seconds. So the moment you say next, the next slide may not be visible to you immediately. Just wait for the slide to come. Thank you very much for your cooperation, but this is how the life is. Ranjan, can you please upload?
Thank you, Pulak. Can you hear me?
Oh, yes.
Okay, thank you. Can I start?
Oh, sure.
Okay, so the title itself speaks that about the parasites. Parasites are smarter than humans. The reason I'm saying that parasites are smarter than humans, because they are constantly giving trouble to our civilization. The parasites are the organisms whose life is dependent on others. They are dependent on the host. So about naming the parasites, I would say there are couple of parasites which I'm going to mention here and tell a few words about them. But as you told me that please say something about the coronavirus. The coronavirus is also a parasite. All viruses are parasites because they cannot survive without the host. So parasites, coronavirus is also a parasite. Bacteria is not a parasite, but fungi is not a parasite, but malaria, trypanosoma, leishmania, they are all parasites because they have to depend on the host. So there are several issues to worry. Malaria caused by the parasite Plasmodium at the tops the list of the global death burden occurring due to protozoan parasites. WHO reported 228 million cases of malaria with 400,000 deaths in the latest malaria report in 2019. Look at that, 280 million cases of malaria. How many cases we are having within 8 months with coronaviruses? Every day you are getting the statistics in the newspaper. Numbers of infection caused by trypanosomatic protozoa like leishmania and trypanosoma are also clinically very, very significant. 500 million residents of the tropical and subtropical regions are susceptible to trypanosomatic infection. And more than 20 million people among the infection-prone population are likely to get contaminated and manifest pathology. So the numbers are very, very high. Although the infection stays asymptomatic, in many cases, fatalities do occur. Tuberculosis is not a parasite, but it's a bacteria. But tuberculosis is one of the causative agents of death in the whole world. Every 100 years, there is a pandemic, occurrence of several diseases, organisms, and that you every day you can see in the newspaper. This is the life cycle of Plasmodium, which is in short, I tell you, Plasmodiums are carried out by the vector Anopheles mosquito. The vector bites an individual, injects the parasite, and the parasites, they find their way in our body, and they harbor they took the shelter in the spleen and liver of the individual, and there they multiply in different. So there is a dimorphic life cycle. Parasite, one human host, and the other in the vector. So next slide, please. So this is the slide for the African trypanosomiasis, more or less the same. It has also the two dimorphic cycle because it is carried by the vector Glossina tsetse fly, and these tsetse fly, they're abundant in the African and South American countries. They bite the individuals, both human and cattle, inject the parasite, and the parasites finds their way to the different places of the body. They're intracellular. They're extracellular parasites. But this beauty of this parasite, trypanosoma, is something which is very, very complicated. And while I was in Zurich, University of Zurich, Switzerland, I was given the task of developing the vaccine against this parasite. But it was not possible because there are several reasons. What are the reasons I will tell you. So next slide, please. Next. See, this is the. I think you have just omitted one slide. Doesn't matter. That is the leishmaniasis. More or less a similar type of transmission through sandfly, which are abundant in our country. There are different types of leishmaniasis. This one of the type is visceral leishmaniasis, which are affecting visceral organ like spleen, liver, etc., bone marrow. So visceral leishmaniasis It is highly endemic in Indian subcontinent and East Africa. An estimated 200,000 to 400,000 new cases of visceral leishmaniasis occur worldwide every year. Now, over 90% of new cases occur in 6 countries like Bangladesh, Brazil, Ethiopia, India, South Africa, South Sudan, and Sudan. You will be surprised to know that I have visited Soviet Russia, and they also have this leishmaniasis. Many people are working on this leishmania. Next. See, this is another form of this leishmaniasis. Leishmaniasis is a collective disease. It is caused by different leishmania parasites. This is a cutaneous leishmaniasis. You can see the sores in the hand. So cutaneous leishmaniasis, about 95% of CL cases occur in Americas, that is South America, the Mediterranean basin, the Middle East, and the Central Asia. Over 2/3 of the new cutaneous leishmaniasis cases occur in 6 countries. What are the countries? Afghanistan, Algeria, Brazil, Colombia, Iran, and Syrian Arab Republic. An estimated 700,000 million to 1.3 million new cases occur worldwide, 216 report, WHO report. So you can see that these organisms, they are very, very rampant, and they are present. They cause the disease, a large number of disease throughout the population. Next. So this is another kind of leishmaniasis caused by the organism, which is called mucocutaneous leishmaniasis. Look at the disfiguring face of this person's mucocutaneous. Almost 90% of mucocutaneous leishmaniasis occur in Bolivia, Brazil, and Peru. So what you can see is that this leishmaniasis is very much occurring in tropical and subtropical countries, including India. And there are different types of leishmaniasis, and you have seen that, and caused by leishmania organisms. Next, please. See, look at this. You'll be surprised to know the fact. Post-Kala-azar dermal leishmaniasis. A patient was affected with leishmania, and he has developed Kala-azar. He has been treated with the drug. Fortunately, we have now drug of all these parasites like Trypanosoma malaria and leishmania. That's why even we are infected, we survive. This post-Kala-azar dermal leishmaniasis is a disease. It comes from a person who has been infected with Kala-azar, and he has been treated with the drug, and he has been cured. And after 5, 6, 7 years later, you can see that the persons come back again to the hospital, and they have developed some nodules in the faces. So those persons who are cured of this Kala-azar, some percentage of that people, they again develop the disease. Therefore, it means that the parasites are not cleared from the body. They still stay inside our body in a dormant stage. Next, please. Next. So this is a letter. It's a very. Can you back? This is a letter. It's a very, very important letter. This letter has been written by one scientist, Charles Doehlemann. He is a medical doctor. He was born in India, and then his father was an ICS, and he was working in the British Indian Army, and then he worked on the disease, and the disease at that time was called black fever, Kala-azar. So he has isolated from the blood, from the WBC, white blood cells, and you could see that there is some cells are there in the third, just like 1, 2, 3, 4, fifth line in the left. That is a picture. He has drawn the picture and he has written that letter, sent that letter to Sir Roland Ross, who was at that time, he was a big man who got the Nobel Prize in 1902. This letter was written in 1902, and at the same time, another scientist, another doctor, young doctor, his name is, next slide. Next slide. Another doctor is William Leishman, and this is Charles Doehlemann. William Leishman is also a British doctor, and he has also been working in India, and he has found out from from a patient, dead patient in Dum Dum. So he was suffering from a fever which was called Dum Dum fever. So that gentleman, he has discovered that parasite, and Charles Doehlemann also discovered that parasite from the which I have shown in the letter. These two persons, they have discovered this parasite, and they wrote a letter to individual letter to Sir Ronald Ross, and then they had a fight. Sir Ronald Ross coined the term to make them, both of them happy, coined the term Leishmania Doehlemann. Leishmania from Leishman, Doehlemann from Doehlemann. So he coined the term Leishmania Doehlemann, the causative agent of visceral leishmaniasis. The picture of another person is Sir UN Brahmachari. Many of you have known UN Brahmachari is the person who has saved the life of millions of people in India who have been, particularly in the eastern part of the country, and some of the in southern part also, and the northeastern part also. These people, this gentleman is a doctor. He has discovered this compound, urea stevamine, is an antimony compound who has saved the life of millions of people. So, and that's why these pictures are very important. Now, next slide, please. See, I'm going to show you the schematic diagram of the parasite Leishmania, a kinetoplastic parasite. What do you mean by kinetoplastic parasite? Any kind of parasite like malaria is a parasite. It has a mitochondria. So some other parasites are also having the mitochondria, but Leishmania is having a mitochondria, and that mitochondria is present in a different way. This is the mitochondria, sir. A pink colored long structure is the mitochondria of the parasite. This mitochondria is very, very big. It's bigger than the nucleus. Mitochondria and nucleus can be separated by subcellular centrifugation, but here mitochondria is very, very big, and you can see on the top of the mitochondria, on top there is a blue region, and that blue region is a specialized region of the mitochondria, and that is called kinetoplast. That kinetoplast is very, very important. It's a site for many, many discoveries. This is the figure on the left side is the parasite, schematic diagram of that parasite present in the vector sandfly, the gut of the sandfly vector. And on the right side, you can see that the parasite in another form of the parasite, because these parasites are called promastigote, and this on the right side, this is called amastigote. The same parasite. Only thing, this round-shaped parasite, which are intercellular, they lose the flagellar structure which are present in the vector. So long, slender, motile promastigotes reside in the sandfly gut, and the round, sessile amastigotes reside inside the host macrophages. Okay. Next slide, please. See, the title of this presentation is "Parasites are smarter than the humans." Why? Look at this structure figure. This is a diagram of a trypanosome life cycle and the variation. Trypanosome is a parasite which is present which is causing the African trypanosomiasis or sleeping sickness in the African countries and also South American countries among the human population and also cattle population. These parasites are carried by the tsetse fly. The tsetse fly, they inject the parasite, bite the individuals, the human and also cattle, inject the parasite, into our system, then what happens? When a foreign organism is being injected into our body, what we do? We always fight. That means we cannot allow the foreign organism to reside in our body. We try to kill them. How we kill? We have the immune system developed already in our body. Our immune system is a system which can destroy the parasite. Now, what happens? You can see 1, 2, 3, 4 color waves are there. What happens when a person is infected with a parasite? This trichoderma parasite, this parasite has the structure of the parasite. I have shown you just like this long slender shaped yellow, green, blue, and pink. And those parasites, they have a surface coat. This surface coat, for example, a person is wearing a blue colored coat and walking in the street, and then suddenly he changes his coat and he's wearing another coat, red coat. So he's now not detectable. So therefore, when a parasite is injecting in our body makes antibody against that parasite. Against the parasite means the surface of the parasite. Surface of the parasite contains a glycoprotein, which is called variant surface glycoprotein. These variant surface glycoproteins at the surface coat, our body starts making antibody against the variant surface glycoproteins. Then what happens? These parasites are so clever, and our body kills all the parasites in our body. Okay, but only a few, 1 or 2 or some of them, they survive. And they again, after surviving, they change their surface coat. The VSG, that variant surface glycoprotein, the nature of the glycoprotein has been changed. So our body is puzzled. So they again start making antibody against that new surface glycoprotein. So then our body again prepares the surface glycoprotein antibody against the surface glycoprotein and kills all the parasite. Beauty is that some of the parasite will still change their behavior, change their coat, and they will wear another coat, and that is called VSG3, variant surface glycoprotein third, this one. So that means what I'm saying is that a parasite after infecting a human changes its behavior at least 100 to 150 times. That means whenever you are being infected with the parasite, you are trying to, your body is killing the parasite. The parasites survive. They change their coat and they again multiply. That's the way. So when I went to University of Zurich, Switzerland, in the year 1979-80, I was told that you clone the gene. At that time, recombinant DNA just came up. And I was told that you please clone the gene for variant surface glycoprotein. So I did the cloning of one of the variant surface, not one, but 2, 3 variant surface glycoprotein gene, and then while doing the sequencing of those genes, we found that those genes are different. That means these variant surface glycoprotein genes, they have changed. So that's why this is the immune evade system of the parasite. They're so clever. They survive inside the host by making fool of the host. So next slide, please. Next slide. So I'm just giving just the mechanism. Mechanism is that this variant surface glycoprotein, I told you, there are 100 to 150 times they can change, and those 100 to 150 genes, they are present inside the chromosome. So what you can see that green, pink, yellow, indigo, and other color, blue color. So these are the different genes present in the chromosome of the parasite, and they're suppressed. Whenever necessary, they change from this place, they jump to the expression site of that organism, of that chromosome. So they change their chromosome expression site. So what I mean is that they can evade the immune system by constantly changing, constantly expressing their genes from one place to another. That's the way they change their behavior. So they are very smart because they can change. Now next slide, please. So this is the picture of, as I showed you, I've shown you the schematic diagram, and this is the real microscopic picture of the parasite Leishmania. This is the promastigote that is present in the sandfly vector. And this is the amastigote. Look at that. There are two big cells are shown here, and two big cells are containing the nucleus, dense colored, and surrounding the nucleus there are small droplets of the organism, this amastigote organism. That means the parasites are coming into us, are being infected by sandfly. The sandfly injects the parasite into our body, and then they multiply. They go to the target organ, which liver, spleen, and different other organisms, and they survive there. They can withstand the detrimental environment of the macrophages. Macrophages are phagocytic cells. Whenever they get a foreign organism, they engulf and they kill it. But here, the parasites cannot be killed. They survive. That's why they maintain their life inside our body. Next slide, please. So now you can see that the parasites are so clever, they can stand like malaria. Malaria is also having, whenever a person is infected with malaria, you give him drug. Fortunately, malaria is having drug. 14 years ago, I went to do postdoctoral studies to work with a person who was involved in the development of malarial vaccine. But today, still malaria vaccine is not yet, has not come out. So malaria, trypanosoma, leishmania, no vaccines are still available, but drugs are there. In my laboratory, for 4 decades, we have been working on basic DNA enzymology of type 1 and type 2 DNA topoisomerase in leishmania, clinical therapeutics, programmed cell death, diagnostics, and toxicity-associated DNA damage and repair. I will tell you about the only 2 DNA topoisomerase. Go back. DNA topoisomerase in leishmania and clinical therapy. What are DNA topoisomerases? From the name, you can see that the enzyme which are involved in changing the DNA topology, they are called DNA topoisomerases. Next slide, please. These enzymes, they work on the DNA, topology of DNA. They change the behavior of the DNA. Whenever a DNA is undergoing either replication or transcription or integration, this DNA has to unwound. And from that, this unwinding is done by the DNA topoisomerases. So DNA topoisomerase is a key enzyme for any kind of DNA transaction processes. So DNA topoisomerase is very important for survival of the organism. Next. I will show you. This is a picture of the kinetoplast DNA network. This kinetoplast DNA is the DNA present in the mitochondria of the parasite trypanosoma and leishmania. This is absent in any of the organism. This is present only in trypanosoma, in malaria, in leishmania, and Critidia, which is a plant pathogen. It's not present in malaria. So this is a unique structure of DNA present in the mitochondria of the parasite. Next slide, please. See, one parasite, whenever they undergo asexual multiplication like bacteria, they are not, although they are eukaryotes, they are small creatures, but they undergo asexual multiplication. That means one parasite will multiply into 2, 2 parasites will multiply into 4, and then if a parasite once multiplies, it all the constituent of the parasite, like the nucleus, the nuclear DNA, the mitochondrial DNA, they have to be replicated, and not only replication, with precision. One, and you can see that a periphery of the kinetoplast DNA picture is shown here. It contains so many small circular DNA molecules. You know how amazing is that? One circular DNA network of the mitochondrial DNA contains 10,000 small circular DNA molecules. 10,000. These 10,000 is only 1 kb long. 1 kb long is not sufficient for coding a protein. So why nature has provided such a huge amount of DNA inside the cell, inside the mitochondria? That was our question. So therefore, one parasite, if wants to multiply, this entire kinetoplast DNA network has to multiply. And if it wants to multiply, then the individual circles should come out from the network. They should multiply, then go back to the network. And that is the model was proposed, and that model was established. So the enzyme which is required to take out the circle from the network and then undergo the replication, bring back to the network again, they are called DNA topoisomerases. Release and then reattachment, go back to the nucleus. That is the property of that, and we have for the first time done this work. DNA topoisomerases. Yes, go back. Next. DNA topoisomerases are called wonder enzymes. They are also called sales magician. What a magician do? Magicians can do both the things, good and bad. They can impress you, and also they can cheat you. These DNA topoisomerases are sales magician because they carry out all the cellular processes involved in vital cellular processes, replication, transcription, recombination, chromosomal segregation, everywhere. They are involved in the interconversion of topological isomers. So these enzymes, which are important in cellular processes, they can be made detrimental to the cell with the help of some of the compounds. So there are two types of DNA topoisomerases. They are called type 1 and type 2. Depending on their mode of action, they can be separated, they can be distinguished. Next. These DNA topoisomerases, they carry out the interconversion. Look at the picture. And there are 4 electron-microscopic pictures of DNA, supercoiled DNA. And look at this at the bottom is a highly entangled DNA. The bottom one and the top one, and in between, all are the same DNA, same, no difference, same DNA, but in different topological state. And if you take those DNAs and run an agarose gel and stain with ethylenedione bromide, you can see that the topological ladders are there. So if you take the DNA, supercoiled DNA, and do the electrophoresis, stain with ethylenedione bromide, you can see on the extreme left the DNA which is giving fluorescence. And in the middle one, you can see the topological isomers, the bands. So you take the DNA, add DNA topoisomerase, and then they will unwind the DNA and make the topological isomer, and it is this. So that is, the assay is very simple. We are doing the assay for these DNA topoisomerases because these DNA topoisomerases are the targets for development of therapeutics. Next. Maybe I will take only 5 more minutes. These are the catalytic activities. I'm not going to tell you again. Next. So this is the major discovery from our laboratory is that I told you DNA topoisomerase 1 and DNA topoisomerase 2. DNA topoisomerase 1 is ubiquitous, is present everywhere throughout the evolution, starting from virus to animal. But the parasites, and they are a single protein. You can see in the top, a human topoisomerase 1. At the left, N-terminal, which is green. In the middle, blue, which is the code domain. And in the C-terminal is red. So it is the active site, tyrosine, is denoted by Y723. This is the enzyme active site. And you can see the next, these two are the DNA topoisomerase, one from Leishmania and another, the second subunit. So human topoisomerase and all other topoisomerase throughout evolution are a single subunit. And here in Leishmania and Trypanosoma, they are the kinetoplastic parasites, they have bisubunit. That is the discovery. So using the fact that this bisubunit enzyme, how it can be tackled, how it can be used to develop drugs. Next. So we have done the localization of the subunits. This is the structure of the parasite, and we can see that they're present in the nucleus, and these two subunits are transcribed and translated into protein, and the proteins are transported into the cytoplasm. They join together and make the hollow enzyme, and they are distributed to the different places. Next. This is what I would say that our laboratory is involved in the biochemistry of DNA topoisomerase as well as the drug development targeted to this enzyme. So we have developed many compounds which are DNA topoisomerase inhibitors, and they have the potentiality to act as anti-leishmal agents. One of my students, very enthusiastic student, the compounds what we have developed in our laboratory with the help of the chemistry students, chemistry faculty members, two types of compounds we develop. One is synthetic compound and the other one is natural compound. You look at the picture of the flower. Is from Taberna montana coronaria. It's a Togo flower in India is abundant, and our chemist friends they have isolated a compound, indole alkaloid, vocamine. This indole alkaloid structure has been determined. This compound is an inhibitor of DNA topoisomerase. The assay is very simple, as I told you. When you run the DNA in the gel, it will at the extreme left you can see that is denoted by SM, means supercoiled monomer. Then you add the enzyme, the DNA structure will be changed, and you will see in lane 2 and 3 that topological isomers are produced, and then you will add a CPT, means cantothysin, is a generalized inhibitor of DNA topoisomerase 1. Reaction is inhibited, you cannot see the topological isomers. But that means it works, and then you are adding vocamine. Vocamine is the compound. You can also see that the topological isomers are not there. They inhibit the topoisomerase reaction. So I have established, and we have published this one. So this next please. We have developed several anti-leishmanial compounds in our laboratory. Some of them are synthetic, some of them are natural, like indolyl quinolin, diospidine, luteolin, abundantly available from our food. So these are the compounds we have developed in our laboratory. Some other compounds are also there, which I did not mention here. So our present status is these compounds, the topoisomerase have been characterized. It has been established as a drug target. Like you know that when a child is infected with diarrhea, he is given the Gramonate, which is nothing but a nalidixic acid. Nalidixic acid is a DNA topoisomerase 2 inhibitors in bacteria. So topoisomerase is an anti-cancer, antibacterial, antiparasitic target. So that's the work we have done. And next. This is a very interesting story. The story is what I say that spread of leishmaniasis from old world to new world. You can see 4,000-year-old Egyptian mummy. You know the PCR is a very powerful technique. This PCR technique, with the use of PCR technique, Leishmania kDNA has been amplified from a 4,000-year-old mummy, and you can see that the positive. So what I mean is that Leishmania is not a new disease. Leishmania is present since 4,000 years. So ancient 800-year-old mummy from New World showing dermal leishmaniasis. So therefore leishmaniasis is present today. They were present 4,000 years before. And then last slide, I guess, is that these are published work. Next slide. Yeah, this is very favorite slide. Pulab told me that it is our subject. Is is a dinosaur, and evidences are there that a fossil has been stand 5 fossil has been taken out, which is the age has been known, and from the stand 5 fossil, it has been found that the fossil contains in the gut of the sunfly some blood, and that blood is from that dinosaur, and that has been stained with the staining staining procedure, and it was found that this is present in the fossil of that sandfly. So this paper was published in 2004, indicating that no, leishmaniasis is not a new disease. It was present, and probably the dinosaur. This is just as prediction. Probably the dinosaurs became extinct by the infection of the leishmaniasis. So that is the story. Next. This is my group from long back, and you can see in the top at the next top left is me. That was 30 years ago, and this is me today at the sitting here. Next, and thank you very much. So this is me. Maybe a couple of years ago. And all my students, they're very well established. And thank you. I'll be happy if you have any questions. But I didn't just mention a few words on the leishmaniasis. Thank you very much.
Thank you, Hemanta. Beautiful lecture. No wonder why you have won so many accolades. I heard or I realized that human look can be deceiving after listening to both of you. Now I am convinced that microbes are more deceiving than the human look. Thank you very much for illuminating lecture.
And one thing I tell you, don't get scared with the coronaviruses, because just I give you statistics. Every day in India, 30,000 people die. 30,000 with some diseases. Natural death, 30,000 per day, every day. And every day, 70,000. Birth is 70,000. So don't worry. Corona, so far you know how many deaths is there in India and also globally, which is nothing compared to all these infectious diseases. So you try to stay yourself in a disciplined way, and then it will be fine.
Thank you for your positive note, Hemanta. Without sparing any more time, let me go to the questions. We have a lot of questions. No wonder why the number of questions will be high. I will try to take as many questions as possible. First, I take our first question, which was asked by Vikas Srivastava to Gunther. Gunther, can you hear me?
Yeah, I'm here.
The question is, would this fungi infect the human beings also through touch or eating? This is one part. Second part is, are these pathogens found in the home plants too?
Well, yes, no, and yes. There are, of course, fungi that infect humans. There are also plant pathogenic fungi that can infect humans, but mostly this is in immunocompromised people. So fungi that are evolved specifically to cause disease, like the smut fungi in plants, they will not infect you, also if you eat them. But there's different examples. For example, Cryptococcus. Cryptococcus is known as a human pathogen, and this is also known that it grows in plants, and it even completes its life cycle in plants. So that's also known. But the very specific interactions are really adapted to a certain host and will not jump between kingdoms usually. Yeah, but in your home plants, I don't know what home plants you guys have. But if you keep your eyes open in your surrounding, you will find pathogens like rust fungi, smut fungi. You find them everywhere in almost all kinds of plants. Yes.
Thank you. Our next question is from Molloy Das. What determines the timing of release of the effector proteins? Am I clear to you?
Yeah, that's a very good question. And that's a question which I cannot answer fully because it's not fully understood. So what is clear is that pathogens recognize certain cues of the host. For example, a plant pathogen recognizes physical and chemical properties of a plant surface, hardness, hydrophobicity, or different molecules. And these molecules will be perceived and they will trigger developmental change, and they will also trigger the activation of virulence factors. This is required for the invasion of the pathogen. However, how the pathogen senses exactly in which cell type it is or in which organ of a host, and this is true for both plant and animal pathogens also, this is not understood on the molecular level. And I'm happy for everyone who is motivated to contribute to answer this because we also try.
The next question came from Shostika Mukherjee. The question is, how much time will it take before the plants become naturally adaptive and immune to these microbes?
Difficult to answer. It depends on what exactly a combination you mean. So in a natural context, you always have a co-evolution between a microbe and a host. That means throughout the evolution, we have virulence factors that adapt, and we have the immune system that adapts. So and you have a genetic variability which reaches from resistant to very susceptible in the natural environment. This is why in the natural environment, you will always have a pathogen being able to infect or cause diseases on some individuals, but not erase the entire species, because otherwise the pathogen would also be extinct. The situation is fundamentally different in agriculture, because what we do is we have a very narrow genetic spectrum and we plant monocultures of wheat, barley, rice, whatever. And if here a pathogen comes in, it will be completely, it can cause an epidemics like we see, because the host plant is not adapted to be resistant. And since the pathogens are much faster in evolution, because they have a much higher generation cycle speed, the plant basically has no chance. So if we want to protect our plant in agriculture from pathogens, we have to help them. We cannot wait for evolution, because we would die by hunger before. So that means we either have to treat the plants with chemicals, or we have to modify the plants by breeding or do GMO plants. This is the two options we basically have.
Gunther, the last question to you is from Udayan De. His question is the range of favorable temperature more or less same for all microbes?
No, of course not. So plant microbes are adapted to their environment where they are. And if you have a microbe, for example, the corn smut, corn is a plant that is from Mexico, and the corn smut pathogen is adapted to high temperatures between 25 and 35 degrees. While if you have a related pathogen species that is adapted to infect barley, you will have an optimized growth range at about 15 to 25 degrees. So they are always adapted to the environment, and they will adapt when they change the environment. much faster than the host.
Thank you, Gunther. Please stay with us for a few more minutes and let me turn to Professor Majumder. Well, Hemantoda, can you hear me? Hemantoda, are you there?
I'm there.
Hemantoda?
Yes, I'm there.
So I start with a sensational question. I don't know, I mean, whether you will like to answer that, but the question came from Amitabh Gupta. The question is, if the coronavirus is a man-made virus, can it as much as a natural virus in outsmarting humans?
There's a typo. Can it be? Can it be as smart?
Can it be as smart?
First of all, this coronavirus, SARS-CoV-2, whether it is a man-made virus or not, that's the question. Because there are some other viruses, SARS-CoV, which was earlier there in the world, MERS, SARS, they are all there, but whether they're artificially created, that's not known. That question cannot be answered. And this
leave it there, because I mean, you know, this is some question. This question is very interesting but very difficult to answer, particularly in the open forum, I guess.
Yeah. Lots of discussion is
Amitabh, I request you to get in touch with Hemantoda.
Yeah.
What about the second part of the question? There is a second part of the question.
Yeah, but the second part of the question is, in other words, does a man-made virus, if there exists one, lose its virulence after some time? Amitabh, let us leave it like that. I mean,
I can answer, but
no, Hemantoda. I mean, that this may not be the right forum. I mean,
okay. Because
you know, our audience from different countries. I don't think that we should pursue. But question is, both the questions are very, very interesting. And you please contact Amitabh. Please contact Hemantoda. He will talk on these two questions. Now, the question from Shati again. And this question is to Hemantoda. If the virus depends on the host for their survival, so in that case, why are the diseased bodies of the COVID victim disposed with high sanitary precautions? Hemantoda, am I clear to you, or should I?
Yeah, it's very clear. But one thing, my major topic of presentation was parasites are more smarter.
No, you can skip the question as well, but yeah. I mean, if you can give a quick answer.
So actually, I would be very, very happy if you ask the questions on the parasites, the real parasites, the protozoan parasites. I can answer this, but that will be different. See, the answers will be it may not be satisfactory to everybody because it is a debatable question. Isn't it?
Extremely debatable question, but this is a valid question as well. But again, I mean, it doesn't fall under your broad topic.
Yeah.
So, Swastika, I again request you to contact.
You can contact me. You can contact my answer.
Majumder, he has the answers, but this may not be the right forum to clarify everything.
No, I could have been given a corona lecture because I published an article in Bengali newspaper. You can read, but this is actually not the. I've been told to give a lecture on the.
Well, Hemantoda, I mean, she has one more question.
Yeah, please.
Which, you can give a short answer. So, is this what we call genetic mutations?
Yeah.
Swastika is asking another question.
Listen, genetic mutation, as I told you, see, these coronavirus, they have been sequenced. 29.9 kilobase long positive stranded RNA virus. And there are, after sequencing 3,000 of the sequences available, it can be classified into 11 groups. That has already been reported. So continuous mutations are also there. Okay. So if the continuous mutations are there, it will be difficult to be handled. So mutations are there. Natural mutations are already there. Even even in case of trypanosoma, they change. These are also called mutations. They change their gene from one place to another region of the chromosome. That is also mutation. Okay, that is also called. Those mutations are not a point mutation. Those mutations are a big jumping gene cases. Okay, but this is a maybe point mutation, maybe some deletions, but the 29.9 kb long RNA is there, and they are coding for 4 different proteins. Those things are all very well known. Okay.
Well, Professor Majumdar, Hemantoda, I know that you have to join another meeting. So formally, yes. I conclude today's technical part. But before that, I express my sincere gratitude to Gunther and Hemantoda from the bottom of my heart. You spared your valuable time. You enriched our knowledge, and we hope that we have had a wonderful evening. And also thanks to my audience, the person who sought questions. I'm sorry I couldn't take all the question. I request that the emails of Professor Majumdar and Professor Doehlemann are with you, or you can contact Shushmita for their email. So whatever questions you have in your mind, you simply go for direct communication if there is not a protocol problem for Shushmita. I mean, I'm a university teacher. I'm not very conversant with the protocols. If there is no problem with the protocols, I mean, many participants may like to contact with the speakers. Thank you very much. With these few words, I hand over to Shushmita again. Shushmita.
Thank you. Before we conclude the session, on behalf of the German Consulate, Kolkata, I would like to thank our guest speakers, Professor Gunther Doehlemann and Professor Hemanta Majumder, for such interesting and informative lectures. Thank you, both of you. I would also like to thank the moderator, Professor Pulakshan Gupta, and the participants for joining the session. As you know, this session of science lectures is available on YouTube at the channel of the German Consulate General, Kolkata. Thank you, Doehlemann Ganguly, for putting the session on YouTube. I would now like to express my gratitude to 3 former Humboldt Fellows, without whose support these web-based science lectures wouldn't have been possible. They are Dr. Shyam Gupta of ISAR Kolkata, Professor Ranjan Ganguly, and Professor Pulakshan Gupta, today's moderator. Both are from Jaipur University. Our next science-based web-based science lectures will be on 5th of November. Please look for the invitation. Now we conclude this session. Thank you and good evening.